everyone, I'm Dr. Pooja. Very warm welcome to MedSpark. Now, today we are going to discuss a very easy but complex topic. Once you got the concept, it is going to be very easy to understand as well as to solve many questions. So, that topic is nothing but the countercurrent mechanism. Countercurrent mechanism. Now, understand how we are going to proceed in this topic. First of all, we are going to have an overview like what is actually happening. I'll make you understand, and then we are going to go through the genome. Okay, so that's how we are going to deal with this topic. So let's start without any further delay. Now, when I say counter current mechanism, there are two words over here. First is the counter, and second is the current. Current in the body, it suggests the flow, and counter means opposite. So there is something going in opposite, and that is nothing but the flow, right? So there is a flow in the opposite direction, which is actually causing this mechanism. So let's understand that. First of all, you should know what are the prerequisites for this counter current mechanism. There are 5 prerequisites which are important in this topic. Nobody is going to tell you that, but they are there. So first of all, whenever you are writing the answers in the prof, you have to write everything, right? So counter current mechanism, what are the prerequisites? What we require to have this kind of mechanism? First thing, there should be two vessels, right? Two vessels which are parallel to each other, and they're having close proximity, so close to each other, parallel to each other, close to each other, but they should have opposite flow. So they should have opposite flow. Opposite flow means opposite direction of the flow. Okay, and the fifth one, and that is the differential permeability. Differential permeability. So these are the 5 prerequisites we are going to require for this mechanism. First is the two parallel vessels close to each other, but they should have flow in opposite direction, and understand they should have differential permeability. Okay, so these are the prerequisites, and these prerequisites are very well met by the nephrons, that is nothing but the tubules in the loop of Henle. So you can see over here just a diagram. Don't get scared, there are many things going on, but one by one we are going to go in the details of everything. So first of all, just focus over here that this is the loop in which one tubule is going down. That means it is the descending limb of loop of Henle, and this over here after the loop it is going to go up. So that we call it as ascending limb of loop of Henle. Okay, but with that we do have vasa recta. Okay, so vasa recta, look over here, it's a part of peritubular capillary, or you can say that these are the branches of peritubular capillary. So vasa recta, one limb of the vasa recta is going down, it is also forming a loop, and one limb is going up, right? And this diagram is actually worth having a look at. Now look over here, these two tubules of the nephron, they are forming the countercurrent mechanism, right, with the tubule itself as well as with the vasa recta. Okay, so first of all, let's discuss what is the site. After the prerequisite, let's discuss where we can find this countercurrent mechanism. Obviously, we are discussing in the kidneys, that is nothing but the nephrons, right? With that, we do have the countercurrent mechanisms in the skin, in the GIT, in the testis also, as well as the research also says that they are present in the lungs and retina. Why I'm writing in the bracket? Because they are not mentioned in the genome or Guyton, right? But yes, there are some books in which they are mentioned, and the research also proven that these countercurrent mechanisms are also present in these two organs or the sites, right? After the site, we are going to discuss about the aim of this countercurrent mechanism. In the kidney, why we require the countercurrent mechanism? There are different aim in different site. today we are going to be precisely talking about the nephron, right? So what is the aim over here? We have to create the medullary hyperosmolarity or hypertonicity. Now understand, our body is made up of 60 to 70% of the water, right? And we cannot afford to lose much of the water via the urine. So that's why we have to conserve the water, and to have that conservation of the water, we require something known as medullary interstitium. Interstitium hypertonicity. So just follow me whatever I'm writing. Medullary interstitium. Which medulla we are talking about? So if I draw a kidney over here, so suppose this is the kidney, right? And suppose this is the ureter coming out from the kidney. So if I divide the kidney into two parts, the outermost part will be called as cortex and the inner part will be called as medulla. Now understand, this medulla can be divided into another two parts. The part of the medulla which is near to the cortex, we are going to call it as outer medulla, and this part over here which is the innermost part, we are going to call it as inner medulla. Inner medulla. Okay. Now if I draw the diagram something like this, so suppose this is the diagram, okay, of the kidney or cross section of the kidney. Suppose this is the cortex, right? This is the medulla, and in the medulla we are going to divide that into the outer medulla and inner medulla. Okay, outer medulla and inner medulla. Now understand, there are two types of nephron. The one type of nephron that is going to be having a loop like this, and one type of nephron which is going to have the loop which is going deep into the inner medulla. If I have to show the diagram which is given in the along, this will look something like this. Now focus over here, there are two different types of nephron. Suppose this is the cortex which is shown, the cross section. This is the outer medulla, this is the inner medulla. Okay. Now there are two types of nephron depending on their length of the loops. Now look over here. One nephron, it is having a shorter loop, right, which is hardly going in an outer medulla, right? But the loop of this nephron is going deep into the inner medulla. Now, understand, as we go down into the inner medulla, the gradient is going to increase in the medullary interstitium. What I mean by that? Let's say over here, the tonicity of the medullary interstitium is 300. Let's say over here it is 500, 800 over here, 1000 over here, and the maximum is going to be deep into the inner medulla, that is going to be 1200. So as we are going down, this osmotic gradient is increasing. That means as the loop is going down, it will help us to conserve more and more water. Okay, and these type of nephrons, since they are going deep into the inner medulla, they are called as juxtamedullary nephrons. Okay, on the other hand, these type of nephrons which is having most of the parts in the outer side, that is cortical side, they are called as cortical nephrons. Understand, 80 to 85% of the nephron, they are the cortical nephron, while 15 to 20% of the nephron, they are juxtamedullary nephrons. That is nothing but which are near to the medulla. Okay, juxtamedullary nephrons. Now, if you understand that, we can move ahead. Now, see over here what is happening. Just now we discussed about this gradient, right? And same is shown over here. As we are going down into the inner medulla, we are going to have more osmolarity. Okay. Now, let's draw with me the diagram, and then you'll understand how this is happening. Okay. So over here, let's say this is the descending limb of loop of Henle, right? It is going to be thin and thick. I'm just drawing over here thin one, right? And in the ascending, I'm going to draw the thick one also. Why, you'll understand in a minute or so. Okay. Now let's continue this. This is the DCT, right? And let's say this is the collecting duct. Okay, now understand what happens. Suppose I said that over here it was 300, 500, 800, 1000 and 1200. Like that, the osmotic gradient is increasing. Now, this descending limb of loop of Henle, it is permeable to only and only water. That means it is going to throw out the water into the medullary interstitium, right? So, as soon as, let's say, isotonic fluid came into this, right, which had the osmolarity of 300, right? Now understand, this 300 fluid came, but outside it is 500, and this tubule is permeable for water. So what is going to happen? The water is going to move out into the medullary interstitium, right? And until what time? Until it becomes 500 inside also. Now this 500 osmolarity fluid comes down over here. Now it is 500 inside the tubule, but outside it is 800, right? So what happens after coming over here? Again, the water will move out to maintain the osmolarity inside and outside at the equilibrium. Okay. Now, until unless it becomes 800, the fluid, that means only and only water, moves out into the medullary interstitium. Now, this 800 osmolarity fluid, it comes down, but what happens as soon as it started to come down outside, again the osmolarity has been increased, that became 1000, right? So again the water moves out. The same thing happens over here unless and until it becomes 1200. Okay. Now, as it goes into the ascending limb of loop of Henle, remember it is permeable for the solutes and not for the water. So there is all the prerequisites met over here. Understand, these tubules are close to each other, parallel to each other. They are having opposite flow, right? As well as they're having differential permeability. What I mean by that? The descending, how to remember? Descending limb of loop of Henle drinks the water. Just remember, descending limb of loop of Henle drinks the water. D for D, that means it is permeable for water and it is almost impermeable for the solutes. So solutes cannot come out of the descending limb of loop of Henle, right? Moving to the ascending limb of loop of Henle, A. Remember, it ate solutes or salts, right? So 8 solute, that means it is permeable for the solutes only and it is impermeable for water. Now the reverse thing is going to happen as we're going to go up, right? So suppose over here, the when the tubular fluid came over here, it was 1200 osmolarity. But outside, when it is going to come over here, outside it is 1000. But it is not permeable for the water. The water cannot come inside. So what should happen now? The solute will move out, right? So over here also the solute is going to move out. So I'm going to show it in the green color. So solute, that means mainly the sodium and the chloride moves out, right? But this is, understand, this is going on passively in the thin limb of loop of Henle. Okay, it is happening passively. We are not talking about the pumps over here, right? Yes, the same thing is going to take place over here. It becomes 1000 as soon as the solutes start to move out. It comes to the equilibrium, but the fluid is still moving upward, and as soon as it comes upward, outside it is 800, inside it is, let's say, 1000, but outside it became 800. So more solute goes out, and as soon as it comes in the thick ascending limb of loop of Henle, we do have a pump which is known as sodium potassium 2 chloride, or we call it as NKCC pump, that is sodium potassium chloride co-transporter, right? So what is going to happen with this pump? Sodium, one is sodium, one potassium, and since they are two positively charged ion, we are going to throw out the 2 chloride out from the tubular cells into the interstitium. Okay, so sodium potassium chloride co-transporter, with the help of this is a pump, understand, and whenever we are going to talk about the pump, that means it is using the ATP, or that's why all the ATPases are called as pumps, okay, or vice versa. The pumps are called as ATPases, okay. Now understand, there are two things happening. First of all, I never mentioned any pump in the descending limb of loop of Henle when I was talking about the water, right? But in the ascending limb of loop of Henle, I mentioned the sodium potassium chloride co-transporter. So that means the water, if I talk about the movement of the water as well as the movement of the solute, what I can say is that the movement of solute is much more as compared to the water. Why I'm saying so? Because understand, the water moves out only via the diffusion. That means it is the passive process. Okay. On the other hand, when we talk in terms of solutes, yes, in the thin ascending limb it is passive, but understand we are also taking the help of the NKCC, that is sodium potassium chloride co-transporter, and that's why the movement of solute out into the medullary interstitium is much higher, and that's why the medullary interstitium, whatever osmolarity it is having, it won't get diluted. Okay, but yes, the continuous movement of the water is occurring into the medullary interstitium. We do not want it to get diluted. For the help of it, the vasa recta also comes into play. That we'll talk about it. So what you have to write about when you're writing in the prof? First thing, you have to write about the gradient, right? So first thing that you're going to write is the gradient. You're going to draw a diagram which will fill up your half page or one page also. Then you're going to write about the differential permeability. That means you're going to write about the water in the descending limb of loop of Henle. You're going to write about the movement of sodium, potassium and chloride. Why we are writing over here sodium and chloride? Because there are Rome K channels also, which will again do what thing? Again secrete the potassium into the tubules back again, right? But you can write over here. Then you're going to write about the solute movement is going to be much more as compared to the water movement, right? All these things you have to mention about that. And the fifth thing you're going to write is the urea. Now, understand, the Genom says that there are at least two different types of transporters present for the urea, and this is going to be the facilitated transport which is going to take place. Okay, that means it is also the passive type of transport. It is not the active transport. Yes, the carrier protein is used, but the energy is not used. Okay, only via the gradient it is going to move, right? So first thing, over here, Genox says that there are different two types of the channels or transporters which are going to be present. What are the two types of channel? UTA and UTB. Okay, so UTA and UTB, again this A are divided into four different subtypes, so 1, 2, 3 and 4. You have to remember 1, 2 and 3 at least. Okay, now this UTA1 and UTA3, UTA1 and 3, I'm writing as like this. So UTA1 and 3, they are present in the collecting duct. Okay, medullary collecting duct for that matter. Now this medullary collecting duct, what it is going to do? It is going to throw the urea out. Understand why it is going to throw the urea out. The urea is only reabsorbed in the PCT. When it comes in the loop of Henle, there is no transporter in the loop of Henle to move this urea out into the interstitium. Okay, so there is no way of reabsorption of this urea, but the water is getting reabsorbed everywhere. The water is reabsorbing here also, in the we can say in the distal convoluted tubule also, right? Water is getting reabsorbed. So the urea concentration when it comes to the CD, it is going to increase, right? Only place where the urea is getting reabsorbed is the PCT, and around 50 to 55% of the urea is going to get reabsorbed in the PCT. Okay, now understand, as soon as the urea comes out into this interstitium of this medulla, what is going to happen? It is also acting as the osmol, so it will help to maintain the osmotic gradient, and which is making the medulla hypertonic. Okay, now understand, if the urea becomes too much into the medulla, what is going to happen? There are urea transporters which are present over here also. So in the descending limb of loop of Henle and the loop, as well as some in the ascending limb, there are urea transporters which are present. Now these urea transporters are slightly different. They are urea transporter A2. Now what they are going to do, if the urea in the interstitium becomes too much, right, what they are going to do is they are going to secrete the urea back into the tubules. So that's their work. Okay. So now this urea, what is going to happen once it is secreted into the tubule, it is going to move towards the collecting duct back again. Now, depending on how much concentration gradient is available, depending on that, the urea, some of the urea might come into the medullary interstitium back, and most of it is going to get excreted. Now understand, there is a pathway going on of the urea. That means urea is coming out from here. Then if it is more, it is again getting secreted back into the tubule. Then it is moving back again over here. And this is like a cycle which is going on of the urea. And that's why it is also called as urea cycling. This is also known as urea cycling. Okay, so this is important phenomena in the creation of the medullary hypertonicity or the so. Okay, so first thing we talked about this counter current multiplier, right? So all these five things you are going to write into. So if I have to make a counter current, I'm just writing CC mechanism. If I have to divide that into two parts, then I'm going to write it as counter current multiplier. These are the two parts of it, and counter current exchanger. So this is the second part, right? Now the counter current multiplier, that is going to multiply the osmolarity of the medullary interstitium, right? So which are these two? Two things are loop of Henle and NCD, that is nothing but the medullary collecting duct. Okay, so this is the medullary collecting duct. Okay, it is not the municipal corporation, so it is the medullary collecting duct. Anyways, counter current exchanger. Who is the exchanger? Vasa recta. We are going to talk about it also, right? Now what is their role? Aim. What was the aim of this counter current mechanism? To create the medullary interstitium hypertonicity, right? Now, just creation is not sufficient. We have to maintain it throughout, right? So, who is going to create it? The creation is done by this multiplier. It is going to multiply it by 4 times, okay? And the maintenance of the hypertonicity is done by this counter current exchanger, okay? Now, why it is called as a multiplier? Understand over here that whenever the tubular fluid came into the descending limb of loop of Henle, it had the osmolarity almost similar to that of the plasma, that is nothing but isotonic, okay? But when it came over here, it became 1200, okay? And which will create the gradient throughout the medullary interstitium also, and that's why it is known as multiplier, because it is multiplying the osmotic gradient by 4 times, right? So milliosmoles per liter. That is why it is called as multiplier. Okay, let's move on to the vasa recta. Vasa recta is very simple. Now understand, we saw the juxtamedullary nephrons. Understand, when we talk about these type of nephrons, there are going to be peritubular capillary which are moving around them, right? So for example, over here, let's understand this. So suppose this is the afferent arteriole, glomerulus it is going to form, and efferent arteriole. These efferent arteriole is going to form peritubular capillary which are going to surround these tubules, right? And that's why they are called as peritubular capillaries. Okay, there is no problem in this type of cortical nephron. So it is going to move like this, going into the outer medulla and coming back and going to form the renal vein ultimately. But understand, they don't have that much of the length to reach into the deep into the medulla, that is inner medulla. So what they do, they are going to give the branches. They are going to give the branches. Those branches are called as vasa recta. Okay, vasa recta, they are the branches of the peritubular capillaries. Okay, now how these vasa rectas are helping us to maintain the osmotic gradient? Let's see that. So whenever we talk in terms of vasa recta, first of all, we have to understand the path of the vasa recta over here. So let's draw the path of the vasa recta. Suppose this is the nephron. Now, this is the afferent arteriole, which is going to form glomerulus, efferent arteriole, and then the peritubular capillaries. These peritubular capillaries, they are going to wound around the tubules, right? Now, what is going to happen? They are going to go, first of all, towards the ascending limb of loop of Henle, right? And then they are going to form a loop, and this loop is going to then come upwards and go towards the DCT. Okay, so that's how their path is going to be. So first of all, they will come towards the ascending limb of loop of Henle, and then they are going to come towards the descending limb of loop of Henle. You have to understand this thing first. Okay, it is not clearly given in the genome, so there is kind of confusion in the student's mind that how this is happening. But once you understand this phenomena first, it is going to be much easier. Now let's see in the diagram, then you'll understand how this is going to maintain the medullary osmolarity. Now see over here, what is happening? First of all, we talked about this vasa recta over here, that it is going to come down from this side. Okay, now see over here, this is also isotonic fluid which is coming into the vasa recta, right? But outside it is going to have much more osmolarity when it is going to go down. Okay, now look over here at this side, what is happening? Yes, I can say that over here, let's say the osmotic gradient is 500. Now, this ascending limb of loop of Henle is throwing out the salts, right? And remember the vasa recta, both the limbs of the vasa recta, they are permeable for both the things. That means it is permeable for water also and solute also, depending on what concentration is more into the medullary interstitium, it is going to take inside that thing, right? So over here, since the ascending limb of loop of Henle putting up more and more salts into the medullary interstitium, it is going to take the salts, right? And since more osmolarity is there outside, so some of the water osmosis also takes place into the medullary interstitium. Why? Because both the limbs are permeable for both the things, that is water also, solutes also. Solute move inside and the water move outside, okay, unless and until it also becomes equal into the osmolarity, that is nothing but 500. But this 500 osmolarity fluid, it goes down, outside it becomes 600, right? So again the same thing is going to happen. Some of the solutes move in and the water move into the medullary interstitium. Same thing happens over here also, right? Unless and until it becomes 1200 milliosmoles, right? Now the reverse thing is going to happen when it is going to go up. 1200 osmolarity fluid moving up. Outside it is, let's say, 900 over here. Now what is going to happen? Outside it is less osmolarity and inside it is more osmolarity. So the reverse thing is going to happen. The water is going to come in and these solutes are going to move out. Again it will move up, right? It became 600 outside, inside let's say it is 900. So what is going to happen? The fluid is going to move in, that means the water is going to move in and the solutes are going to move out unless and until it becomes isotonic again. This is the picture I have taken from the net. So this exactly does not become 300, it remains 325. There are some laws of the solutes. The genome says it is as when it leaves vasa recta, when it leaves the medulla, it is going to be 325. That we are going to see the authenticated diagram also. So what point I have to make is that the reverse flow is going to take place over here, right? So let's see the genome right now and then we will be talking about these things. I hope you understand that how this is going to take place, okay? Now one more thing just I want to mention over here. You can write the applied aspect. If I have to block this channel, since it is a pump, I can block it via the drug. The drugs which are blocking the pumps in the loop and they're causing the diuresis. So that's why we call them as loop diuretics. Okay, if I block this channel, sodium, potassium and chloride, they will not be able to move outside of the tubule and they are osmoles. So they are going to hold on to the water. So the water is also going to lost into the urine, and that's why the diuresis is going to take place, and that's why we call these drugs as diuretics. Some examples: furosemide, then torsemide, bumetanide, bumetanide. These are some examples of these loop diuretics. Okay, so let's move on to the genome and let's complete the reading also side by side, and then we will have a complete understanding of this topic. So look over here, counter current mechanism. It is given in the chapter 37, right? The page number might vary depending on which edition you are using. Okay, the counter current mechanism, the concentrating mechanism. So first thing you have to highlight, this is the concentrating mechanism depending on the maintenance of the gradient of increasing osmolarity along the medullary pyramids. Okay, so medullary pyramid, that means the medulla that we are talking about, right? So the genome clearly writes over here that the medulla, what is the main function of the medulla? Concentrating the urine. Okay, this gradient is produced by the operation of the loop of Henle as a countercurrent multiplier, and it is maintained by the vasa recta countercurrent exchanger. But understand, later on it also mentions the collecting duct as well, right? The countercurrent system is the system in which the inflow runs parallel to, counter to, and in the close proximity to the outflow for some distance, right? So these are the prerequisites which you have to mention in your exams. Okay, this occur for both the loops of Henle and the vasa recta in the renal medulla that we just now saw. The operation of each loop of Henle as a countercurrent multiplier depends on the high permeability of the thin descending limb to water via aquaporin 1. So you just can add over there are aquaporin 1s. Now, when we talk about in terms of ADH, the ADH is going to cause the activation of aquaporin 2, insertion and activation of aquaporin 2. On the other hand, these are going to be constitutionally present, aquaporin 1, which are present in the descending limb of loop of Henle. Okay. Now, the active transport of sodium and chloride out of the thick ascending limb. Now, understand over here what it is saying. Look over here carefully. It is saying active transport in the thick. In the thin also the passive transport takes place. Okay. And the inflow of the tubular fluid from the proximal tubule with outflow into the distal tubule, which we also discussed. Okay. The process can be explained using hypothetical steps leading to the normal equilibrium condition, although the steps do not occur in the. Okay, so whatever diagram or figure is given into the genome, that doesn't happen exactly those like those steps, but we can understand. It is better to understand. Okay, so that's how they have given this figure. It is also important to remember that the equilibrium is maintained unless the osmotic gradient is washed out, which will not happen usually. Okay, so these steps are summarized in the figure 3715. So let's see in the figure. So first thing over here, this is the thin descending limb, right? Understand? And this is the thick ascending limb, right? Suppose if I say that if there is no gradient, no movement of the water or solute will take place. Okay, so it will not be able to conserve the water, it will not be able to make the medulla as a hypertonic, right? Will not be able to conserve any amount of water. So what this figure is telling us now, over here they are saying that operation of the loop of Henle as countercurrent multiplier producing a gradient of hyperosmolarity in the medullary interstitium, right? It is none other than what we discussed just now. The process of generation of gradient is illustrated as occurring in the hypothetical steps, starting at A, where the osmolarity in both the limbs and the interstitium is 300 milliosmol per kg of the water. We can take it as per liter also, right? The pumps in the thick ascending limb move sodium and chloride into the interstitium. We know that. Look over here, this is moving the sodium and chloride, and that's why if you are moving the salts outside, it is going to be, or in the medullary interstitium it is going to be hypertonicity, and inside, since the salt is moving out, so in the tubule the tonicity is going to decrease. Okay, That's what it is writing as. And this equilibrates with the fluid in the descending limb, right? And however, isotonic fluid continues to flow into the descending limb and hypotonic fluid out of the thick ascending limb. Continued operation of the pumps makes the fluid leaving the thick ascending limb even more hypotonic. So this is one of the questions which comes in the NEET or INICT or any entrance exam, that fluid leaving the thick ascending limb of loop of Henle or ascending limb of loop of Henle, it is going to be hypotonic. Or they can ask you fluid entering into the DCT, it is going to be hypotonic. Okay, while hypertonicity accumulates at the apex of the loop, apex is this one. Okay, so it is going to have much tonicity, higher tonicity, that is hypertonicity, and that is what it is given over here. Now understand, with the help of this diagram, what they are showing, that suppose that continuously I keep on accumulating the salts into the medullary interstitium, finally it is going to form the gradient like this. Okay, and with the help of gradient, I'm going to absorb more and more water, right? And that is the role of the kidney, main role of the kidney. And over here the vasa recta is shown. First of all, we'll continue the reading over here. Summarize in this for the cortical nephron with no thin ascending limb. Okay, assume first a condition in which osmolarity is 300 milliosmol per kg of the water, which was shown in the diagram, throughout the descending and ascending limb and the medullary interstitium. Assume in addition that the pumps in the thick ascending limb can pump 100 milliosmoles per kg of sodium and chloride from the tubular fluid into the interstitium, which is shown in the another diagram, that is diagram number B, right? 100 milliosmole per kg, it is throwing into the medullary interstitium, right? Increasing the interstitial osmolarity to 400. Why? Because it is throwing 100 milliosmoles, right? Water then moves out of the thin descending limb and its contents equilibrate with the interstitium, which is shown in the B, which we discussed. However, a fluid containing 300 milliosmol of per kg water is continuously entering this limb from the proximal tubule, so the gradient against which the sodium and chloride are pumped is reduced and more enters the interstitium. So what it is saying, that continuously the flow is occurring, right? We're discussing very slowly, stepwise, but this is not going to take place in vivo, right? Continuously the isotonic fluid is coming. So that's why this pump is getting activated more and more. And what is happening over here? There is a creation of gradient. Why? Because the pump is continuously throwing the salt out, isn't it? Now, meanwhile, hypotonic fluid flows into the distal tubule. Why? Because we have thrown the sodium and chloride outside into the medullary interstitium. So, inside the tubule, less solutes are present, right? And we know that the ascending limb of leucophilate is impermeable to water, right? So, it is kind of a diluted fluid which is moving towards the distal limb or distal tubule. The isotonic and subsequently hypotonic fluid flows into the ascending thick limb. The process keeps repeating and the final result is the gradient of osmolality from the top to the bottom of the loop is going to be generated. In juxtamedullary nephrons with the longer loop, thin ascending limbs, the osmotic gradient is spread over the greater distance. We know that in the juxtamedullary nephron, it is going to have longer loops, right? And that's why the gradient is going to be spread along the longer length, that is there. And the osmolality at the tip of the loop is greater. We know that it is 1200 also, though it is of vasa recta. I'm just showing it. Okay. Now, this is because of the thin ascending limb is relatively impermeable to water but permeable to solutes, that is sodium chloride. Why we do not talk about potassium? Understand, the potassium come into the medullary interstitium, but there are channels which are known as the Rome K channels, which are renal outer medullary potassium channel. Through that, the potassium again gets secreted back into the tubule. So that's why we do not talk about the potassium over here. Therefore, sodium chloride moves down their concentration gradients into the interstitium and there is additional passive countercurrent multiplication. Okay, this question has been coming in many exams, and some of the books, MCQ books especially, they have given the answer wrongly, that only thick ascending part takes place in the countercurrent mechanism. No, understand, thin part also contributes to it, or to multiplication. It is clearly given in the genome. The greater the length of the loop of Henle, the greater the osmolarity that can be reached at the tip of the medulla. So hence, if they ask the question, who is going to be mainly responsible for the creation of the gradient, then we are going to answer it as juxtamedullary nephron, because they are having longer loop. The osmotic gradient in the medullary pyramids would not last long if the sodium and urea in the interstitial spaces were removed by the circulation, right? So over here, the genome also mentions the urea, right? If you remove that from the interstitial, there will be no gradient. Okay, these solutes remain in the pyramids primarily because the vasa recta operate as a countercurrent exchanger. So again, there is a question: who is the exchanger? We know that it is vasa recta. The solutes diffuses out of the vessel conducting blood towards the cortex. Okay, the genome is shown a bit of the reverse, what it is actually into the medulla, so you might get confused, but there is no confusion. It is clearly writing when it is going down, what it is doing, it is putting the water into the medullary interstitium and taking the solutes in, right? When it is going towards the medulla, right, and when it is moving towards the cortex, it is taking the water in. That means when it was leaving outside into the, yeah, let's say the capillaries are moving upwards, what is happening? They are taking the water in. That means they are not allowing the hypertonicity to get diluted into the interstitium. So what they are doing? They are maintaining the hypertonicity. Okay. So that's what the genome is telling us. The solute diffuses out of the vessel conducting blood towards the cortex, right? And into the vessel descending into the pyramid. Uh, it is going in when it is going towards the pyramid. The solutes are moving out when it is going towards the cortex. That means outside. Conversely, water diffuses out of the descending vessel and into the fenestrated ascending vessel. Therefore, the solutes tend to recirculate into the medulla and water tend to bypass it, right? Water is going out, right? While the solutes are recirculating over there. So, that hypertonicity is maintained, right? So, ultimate aim is achieved. The water removed from the collecting duct in the premise is also removed by the vasa recta and enters the general circulation. Countercurrent exchange is passive process. It depends on the movement of water and could not maintain the osmotic gradient along the pyramid if the process of the countercurrent multiplication of the loop of Henle were to cease. Okay, so please remember, they can ask you the countercurrent exchanger is active, passive, primary active, secondary active, what it is. It is passive. Okay, it is worth noting that there is very large osmotic gradient in the loop of Henle and in the presence of vasopressin, that means nothing but the antidiuretic hormone, that is nothing but the ADH, that is antidiuretic hormone in the collecting duct. Now what the ADH is going to do, ADH will allow more water channels to open, that is aquaporin 2, and that will allow more and more water to come from the collecting duct into the medullary interstitium, which will be again taken up by the vasa recta while going out. Okay. Now, it is countercurrent system that makes the gradient possible by spreading it along the system of the tubule 1 centimeter or more in length. Okay, rather than across a single layer of cell is only a few micrometer thick. So, longer the length, we learn that, and longer will be the gradient, more will be the efficiency, right? That is what it is saying. Rather than having a very small thickness, we do have a longer loop, and over the 1 centimeter, the gradient is going to change. There are other examples of operation of countercurrent exchanger in the animals. One of that is heat exchange between the arteries and vasocomitants. Now, what is vasocomitants? Now, understand, these are present in the skin of the limbs. We wrote the sides, right? One of that was skin, right? So those specific veins which are directly joined to the arteries, right, that are called as vasocomitants. of the limb. To minor degree in the human, but to major degree in mammals living in the cold water. What is the purpose of it? To conserve the heat, since the water is very, very cold. Heat is transferred from the arterial blood flowing into the limbs to the adjacent vein, draining blood back into the body, making the tips of the limbs cold while conserving the body heat. So the heat is not going towards the capillaries, rather it is bypassing the capillaries and going into the veins. Okay, that's what it is telling us. Next. So this diagram we discuss, we do not need to discuss that again and again. Role of the urea, let's discuss that. Urea contributes to the establishment of the osmotic gradient in the medullary pyramids and to the ability to form the concentrated urine. Again, it is helping in the multiplier. In the collecting data. Urea transport is mediated by urea transporter, presumably by facilitated diffusion. It is not the active transport. I told you it is the carrier protein, but not using the energies. That's why it's a passive. And which type of passive? Facilitated diffusion. There are at least 4 isoforms of the transporter protein. UTA is going to be there, that is UTA 1, 2, 3, 4. UTB is found in the RBCs and in the vasa recta, that is erythrocyte, and in the descending limb of vasa recta. Urea transporter in the collecting duct is mediated by A1 and A3, and both are regulated by vasopressin. So you can mention over there that if the vasopressin is present, these two are going to get activated by the ADH. So I can write over here, in the presence of ADH, they are going to get activated, and both are regulated by these vasopressin. During antidiuresis, anti means again, diuresis means the excretion of water. We are going anti to it, that means we are preserving the water. When the vasopressin is high, the amount of urea deposited in the medullary interstitium is going to increase. We know that because it is stimulating these two things. And osmol is moving out. Behind those osmol, the water is also going to move out. So additionally, it is helping us, right? Thus increasing the concentrating capacity of the kidney. In addition, the amount of urea in the medullary interstitium and consequently in the urine varies with the amount of urea filtered. That means whatever filtration, where it is going to occur, it occurs in the Bowman's capsule, isn't it? Suppose this is the Bowman's capsule. Suppose this is the afferent arteriole, right? This is, let's say, the glomerulus and efferent arteriole. So let's say more urea is going to get filtered, then definitely more urea will come towards the CD. If less urea got filtered, less urea will come towards the CD, right? That's what it is telling us over here. Let's move on to the next thing, that is, and this terms varies with the dietary intake of the protein. Many a times this question has been also asked. Therefore, high protein diet increases the ability of the kidney to concentrate the urine, and low protein diet reduces the kidney's ability. Now understand, whenever we are going to eat the protein, it is going to create a byproduct which is nothing but urea, and the urea is going to help us to concentrate the urine. And if we eat more protein, more urea will be created, more capacity of the kidney to concentrate the urine will be developed. Okay, coming to the osmotic diuresis, which is the last part, the presence of large quantities of unreabsorbed solutes, that means the solutes which are more into the tubules, understand that, in the renal tubules causes increase in the renal volume. Why? Because they're acting as the osmoles and they are going to hold on to the water. They won't allow the water to go into the interstitium, called as osmotic diuresis. Why? Because these unabsorbed solute are acting as the osmoles. They will allow the diuresis. I told you just now, the diuresis means excretion of the water, right? So this is because of the osmoles. So that's why this diuresis is called as osmotic diuresis. Solutes that are not reabsorbed in the proximal tubule exert an appreciable osmotic effect as the volume of the tubular fluid decreases and the concentration rises, right? If the solutes are not reabsorbed in the PCT because they are unreabsorbable, right? That is the condition that we are putting over here, right? So what is going to happen? Water is going to move out, And whatever solutes which were not reabsorbed, their concentration per liter is going to increase, isn't it? The amount is similar, but the concentration is increased. Why? Because PCT is going to reabsorb the water, right? So what, that's what they are saying, that the concentration is going to rise, therefore they hold water into the tubule. In addition, the concentration gradient against which sodium can be pumped out of the proximal tubules is limited. Now there is some limit to the sodium reabsorption. Usually 60 to 70% of the sodium is absorbed in the proximal tubule. It's not like that if sodium is going to reabsorb continuously. It is going to have certain limits, right? Normally the movement of water out of the proximal tubule prevents any appreciable gradient from developing, but sodium concentration in the fluid falls. Now understand, we are saying that less water is getting reabsorbed. If less water is getting reabsorbed, obviously the sodium concentration, the sodium is going to get diluted again in that water. So the sodium concentration is falling, right? So whatever gradient was available for the sodium, it is also decreasing, right? So in the fluid falls when water reabsorption is decreased because of the presence of tubular fluid of increased amount of unreabsorbable solutes. The limiting concentration gradient is reached, right? So that is the maximum capacity, right? Or that is the gradient. And further proximal reabsorption of the sodium is also prevented. So if there are more unreabsorbable solutes, that is causing a harm rather than any benefit. Okay, more sodium remain in the tubule and water stays with it. The result is that the loop of Henle is presented with a greatly increased volume of isotonic fluid. More volume is going to come into the loop of Henle since unreabsorbable solutes were there, right? So water was there. Since the water was there, the limiting concentration gradient of the sodium is reached. Sodium also cannot go out. So all three of them is going to go towards the loop of Henle. So loop of Henle is going to meet all three of them, right? Which is going to be isotonic. Let's move on. This fluid has decreased sodium concentration. Amount is increased, but the concentration has been decreased because more water was there. Understand that. But the total amount of sodium reaching the loop per unit time has been increased, right? It is increased. In the loop, reabsorption of water and sodium is decreased because the medullary hypertonicity is decreased. Okay. The decrease is due to primarily to decreased reabsorption of sodium, potassium and chloride in the ascending limb of loop, because the limiting concentration gradient for the sodium reabsorption is reached, which is going to act as the main solute is reached. More fluid passes through the distal tubule, and because of decrease in the osmotic gradient along the medullary pyramid, less water reabsorbed in the collecting ducts. The result is marked increase in the urine volume. Okay, so please remember, because of this what is happening is less water reabsorption and ultimately everything is going into the urine, and that's why the urine volume is going to be increased with the increase in the excretion of sodium and other electrolyte, right? So this is the osmotic diuresis. Now how we can use this information for our benefit? Suppose if some patient is there of hypertension, or let's say cardiac heart failure or congestive heart failure, we do not want much of the volume in the blood. So we want the volume of the blood to be reduced, right? So what we are going to do? We are going to cause this osmotic diuresis. So the preload of the heart is going to decrease. So let's see that how we are using that information. Osmotic diuresis is produced by the administration of the compounds such as mannitol and related polysaccharides that are filtered but not reabsorbed. They will be filtered, but since they are big molecule, they cannot cross the tubules. Okay, it is also produced by the naturally occurring substance when they are present in amount exceeding the capacity of the tubules to reabsorb them. Okay, for example, diabetes mellitus. In the diabetes mellitus, if the blood glucose is very high, which is crossing the Tmax or renal threshold, right? The glucose in the glomerular filtrate is going to be high. Since their high blood glucose, and those blood glucose through the renal plasma flow is going to get filtered into the tubules. So there also it is going to be high. This is known as filtered load. Whatever load came via filtration, that is known as filtered load, will exceed the Tmax or TMG, that is the tubular or transport maximum for the glucose, right? The glucose reabsorption is going to have a limit. Okay, that limit has been crossed. The glucose will remain in the tubules causing polyuria. Glucose will also act as the osmotes. Osmotic diuresis can also be produced by the infusion of large amount of sodium chloride or urea. It is important to recognize the difference between osmotic diuresis and a water diuresis. Now, what is the difference between these two? In the water diuresis, the amount of water reabsorbed in the proximal portion in the nephron is normal, and the maximum urine flow that can be produced is about 16 ml per minute. Now understand what we talked about osmotic diuresis, that the water reabsorption, that was the only thing, right? The water reabsorption in the PCT has been decreased in the osmotic diuresis. In the case of water diuresis, it is going to be normal. So why the water diuresis are occurring? Because understand, more water is coming into the filtration, right? That means if the person is drinking more water, that is going to come into the urine, okay? But it won't affect the other things, okay? Can be produced about 16 ml per minute. So there is not going to be much of the urine production. So let's move on. In osmotic diuresis, increased urine flow is due to decreased water reabsorption, right? In the proximal tubules and loop, and very large urine flows can be produced. So that is the difference. Very large amount of urine is going to be produced in the osmotic diuresis since the water reabsorption is decreased. In the water diuresis, only much of the water is coming in the filter. Whatever is excess amount of water, that is getting excreted. Okay, but it won't be causing much of the urine volume, okay, to be increased. Right, let's move on. As the load of excreted solute is increased, the concentration of the urine approaches that of the plasma, obviously, right? So it will go towards the isotonicity. In spite of maximal vasopressin secretion, because an increasingly large fraction of excreted urine is isotonic proximal tubular fluid, right? So the urine also becomes isotonic. If osmotic diuresis is produced in the animal with the diabetic insipidus, the urine concentration rises for the same reason. Let's move on. Relation of urine concentration to GFR. The magnitude of osmotic gradient along the medullary pyramid is increased when the rate of flow of fluid through the loops of Henle is decreased. Now understand, this is the inverse correlationship which is talked over here. The reduction of the GFR, such as that caused by dehydration, produces decreased in the volume of fluid presented to the countercurrent mechanism, so that the rate of flow in the loops decline. Obviously, GFR is decreased, the volume which is presented towards the loop is also going to be decreased, and the urine becomes more concentrated. When the GFR is low, the urine can become quite concentrated in the absence of vasopressin or source. Please remember that. How this is going to be useful for us? If the renal artery is constricted in the animal with the diabetes insipidus, the urine excreted on the side of constriction becomes hypertonic. Okay, because of the reduction in the GFR, whereas that excreted on the opposite side remains hypotonic. Okay, so that's how we can use this information for our own benefit. Understand, this will not be applicable in the human beings, so nobody is going to ask you this question. Okay, but if you want, if you have time in the profs, you can write that also, because whenever we talk about the urine in vivo, whatever urine is formed by two different kidneys, it is ultimately going to mix in the urinary bladder. So we will not be able to, do this experiment in vivo. Okay, so we won't be able to differentiate between these two things. Okay, now lastly, let's see what are the graphs given over here. Whenever you are going to see any graph, you have to see first of all what is given in the x-axis and what is given in the y-axis. So please look over here, x-axis is solute load and the y-axis is urine flow. It's not the renal plasma flow. Don't get confused between that, okay? So whenever we are increasing the solute load in the tubules, what is happening? Look over here, if it is, we are talking about isosmotic urine, the urine flow will increase, right? Osmotic diuresis, remember, if you increase the solute load, the urine flow will also increase because the sodium also and the water mainly is going to stay in the tubules, okay? So it is having the linear relationship. Now when we talk in terms of the vasopressin secretion, if maximum vasopressin is getting secreted, what is happening over here? So let's take a point over here. Let's say I take a point over here as this 2.7 milliosmol per minute. Suppose I do not have the vasopressin. What you can see, what is the urine flow? It is almost 9 ml per minute. Okay. On the other hand, if we do have vasopressin, you can see it is almost 4 to 5 liters. That means this vasopressin is actually helping us to reabsorb more and more water. Okay, and that's why it is called as anti-diuretic hormone. Okay, by acting on the V2 receptor, it opens up aquaporin 2 and also it is helping us to create the medullary interstitium hypertonicity with the help of urea transporter, right? So that's why we can absorb more and more water. It is helping us to conserve the water. On the other hand, if it is diabetic insipidus, what is going to happen? You can see the urine flow is going to be much increase. Diabetes insipidus, when it is going to happen? Whenever there is an absence of ADH. Usually when there is an absence of ADH or the ADH receptors are not working properly. Okay, so the flow of the urine is going to increase. Let's move on to the other graph over here. That is, urine flow is given on the x-axis now and the urine osmolarity is given over here. Now isosmotic, it is around 285 to 295 milliosmoles per liter, but let's take 300 over here for our convenience. Now if we talk about maximal vasopressin, you can see over here that the osmolarity, as the vasopressin secretion is increased, what is happening? The osmolarity, urine osmolarity is increasing and the urine flow is decreasing. Okay, so we can see that as soon as the vasopressin is increased, right? So this green is showing the increased levels of vasopressin. When the vasopressin is high, obviously the urine osmolarity is also high because we know that it is going to cause more and more watery absorption, right? On the other hand, if we talk about the absence of vasopressin, it is going to cause much higher urinary flow and very low urine osmolarity. Okay, so that is about the diabetes insipidus and that is all about the counter current mechanism. I hope you understood the counter current mechanism very well, right? And I hope you like the session. Thank you very much.